Buenas and half a day to the Hamzu. My name is Vince Fabian. Welcome back. So my final lab project is on assessing the chemoreceptive capabilities of the invasive flatworm, Platydemus manokwari, by looking at the action potentials in the longitudinal nerve cord after chemical exposure. So I chose this organism to study because I wanted to focus my project on something that I thought would help conservation tactics. I previously have seen this worm when I was volunteering at Jigo's Cycad Refuge, and it's there where I kind of learned a little bit about their background and behavior from one of the program managers. So I'm gonna give you guys a little bit of info on the context. So here, Platydemus manokwari is a predatory terrestrial flatworm within the phylum Platyhelminthes, and it originates in New Guinea. Now, this flatworm is actually invasive and is thought to have come in from imported plants through the soil. And because of this, it has resulted in a reduced snail populations in numerous locations. They're pretty effective snail killers because of their ability to track their mucus trails, and they can immobilize their prey using their own sticky mucus. So here in this picture, you can see how this flatworm is able to immobilize its prey, and then it uses its body to, I guess, engulf the snail so that it can't move. And after that, it secretes different chemicals that allow it to digest the snail, and then it uses its pharynx here, pictured as this white tube on the ventral side, to feed on it. Now, because of this history, some locations have thought of using this flatworm as a biocontrol for the invasive giant African land snail, Achatina folica, and one of the places is actually here on Guam. So here on Guam, it actually worked a little too well, and it also involved the killing of native snail populations. So that's why we kind of want to try to get rid of this now. So according to previous literature, snail tracks are commonly used among snails themselves in communication, reproduction, and locomotion. So snails have typically followed tracks of conspecifics or those of the same species, and this implies that the chemical composition of snail tracks may differ from species to species. So this leads me to the question of what specific chemicals are these flatworms sensing, and does this mean that Platydemus can also sense these differences? And if so, does it have a preference, and how do they discriminate between species? So one study actually conducted by Iwai and Sugiura showed prey preference being exhibited in a flatworm choice experiment between four snail species. Now, locally, previous observations made by some of my fellow colleagues who work with snails, as well as Dr. Fiedler, mentioned that Platydemus seemed to favor the native Partulas over the non-native cinnamon bun snails, or the Ganaxus species. And the endangered fragile tree snails, Samoana fragilis, was actually preferred overall. These prior observations kind of led me to the proposed hypothesis that P. manokwari actually does have a preference, and this preference can be recorded through measurements of action potentials within its longitudinal nerve cords. So I was actually trying to find a flatworm for this project ever since we got the instructions for it, but I haven't been so lucky. But fortunately, Dr. Fiedler was actually able to find a flatworm for me just in the nick of time, and I was able to perform this behavioral experiment. So it's actually over here, as you can see. It's a little tiny, like just in this medicine pill. So basically, what I did with my choice experiment was I tried to perform a Y maze, but I didn't have the materials to make it enclosed, so I did a modified Y maze, which is basically just this pill actually in the middle over here, and then the two choices on either side. So the different variables that I actually used in this experiment are the different chemical cues. So I used water as a control. And then as for my snail track, I had giant African snails that I actually got from Asialin. And I also used bleach as a substitute because I couldn't find another snail that I could use to look at the preference. I also wanted to look at the potential speed at which the flatworm moved in response to these different stimuli. Now, before I got into the actual experiment, I decided to do first an assessment because I wanted to see if this phenomenon actually happened within this particular organism. So first off, I tried doing these different stimuli with a little sample on a bottle cap, and then I put it near the end of the tube over here to see if it would respond. And apparently it didn't really have much of a response when it came to the snail tracks. I'm guessing because it wasn't as concentrated, but it did have a response to the bleach. So after that, I decided to use a knife and to just kind of put the snail track a little closer to the flatworm to see if it would move, and it didn't really have much of a response. And then finally, I did a trail in front of it that led to the exit of the pill bottle, and that's kind of how I got it to move out of the tube. As for the results of the experiment, what I noticed was this. So these here are the different choices that I put on either side of the container. And then this is the behavior that I observed, and this is the speed that I calculated. So for the choices, I decided to keep the water and the bleach in the bottle cap because it seemed as though those two were okay being in a bottle cap. Whereas for the snail track, I kept it as a trail because that elicited the most response. So when faced with the choice between water and bleach, it showed that there was no movement for the flatworm, a speed of 0. Whereas when it was given a choice between the water and a snail track, it actually did move towards the snail track in a general direction. But after it got out, it wasn't able to find the source. It just kind of moved towards the general direction of the snail track. And the speed that I calculated was approximately 0.085 millimeters per second. As for the choice between the bleach and the snail track, what I found was that there was initial movement towards the snail track, but then after a while, when it started to sense the smell of the bleach, it seemed to retreat as if it was being alarmed by a predator. And for that speed, it was actually 0.064, which, although is a difference, doesn't actually seem like much of a difference. So moving on to the physiological experiment, I'm first going to give a basic introduction on flatworm anatomy. So for the nervous system of the flatworm, basically they have four different types of, I guess, nerve innervations. So there is the cerebral ganglia, which is basically the integrating center for the flatworm. And then there's these two longitudinal nerve cords, which is characteristic of these flatworms. And then between them are lateral nerve cords that kind of connect these two. And then along the periphery of the flatworm's epidermis, there is a peripheral nerve plexus in which the other sensory organs can be found. As for chemoreception, these sensory organs are actually called auricles. So here, the auricles are placed near the head, just a little bit behind the eye spot. And I put here a little micrograph of what the auricle looks like. It's this tiny little thing over here. And this is connected to the cerebral ganglia where it sends information to tell the worm that this is what it's smelling. So for my proposed experiment, what I'm thinking of doing if I didn't have quarantine was to first anesthetize the worm using a 10% ethanol solution in saline, which I got from the earthworm action potential lab. I hope that it works, but if I find that it doesn't, then I will adjust it. Then after that, I will dissect it here along the middle, along the ventral side. I'm gonna first pin the worm and then dissect along the middle to split open the inside as like we did in the worm lab to reveal the nerve cords. And any waste that I find that I would clean out and then keep the worm well hydrated because it relies on diffusion to be alive. As for the action potential recording, I plan on using LabChart, and I will hook the shielded lead wire micro hooks onto the longitudinal nerve cords on either side, shown here as these black and white pins. And now this is a procedure that I got from the cockroach ventral nerve cord lab. And after that, I'm going to pin the earth electrode onto in contact with the body cavity, but not touching the nerve cord, also stated in the lab. After this setup is complete, then I will record the baseline action potentials under normal conditions to see what it's like normally, and then measure action potentials under different stimuli of the auricle. So I would touch the auricle, see what happens. I would puff the auricle with a pipette, and then I would introduce the chemical cues to see how that changes. And I would, between the treatments, I would rinse the auricle with the saline solution so that no leftover is left behind. As for my conclusions of this study, I found that in the behavioral experiment, the chemoreception in P. manokwari is observed, and it can discriminate between the potential prey and threat. So the threat being the bleach, and the prey being the giant African snail trail. As for directionality, it's a bit general, but it does not exhibit the capability to find the source. It potentially uses this approach to kind of increase prey interaction, but it won't actually look for it. The concentration may be a factor, as can be seen by the different assessment methods that are used. As for the significance, research into the chemoreceptive capabilities of this flatworm may help create potential conservation strategies, for example, chemical luring or deterrence, as well as identifying targeted species. Characterizing physiological responses of the sensory system in flatworms may contribute to the ongoing research of invertebrate chemoreception, which is still being researched. Understanding this mechanism behind this response may also explain the success of this species. For further research, I would try to maybe look into isolating the specific chemicals within the snail tracks and observing the effects of that stimuli on the flatworm physiological response to see if we could find out if this is a combinatorial chemoreception. Thank you.